In this topic, we're going to have a look at translation. So we're going to look at what is translation, what is amino acid activation, and then the different steps in translation. So you've got initiation, elongation, and termination. Remember that messenger RNA is made from DNA in a process called transcription. So we had a look at this in the last topic. So transcription occurs inside the nucleus. The messenger RNA then moves out of the nucleus and into the cytoplasm through the nuclear pores. Together with tRNA and ribosomes, messenger RNA is translated into a sequence of amino acids, which will form a protein. So what is translation? Well, this is the process by which the messenger RNA, which you can see here as the black strip, from the nucleus of the cell moves into the cytoplasm and it forms a polypeptide in accordance with the sequence of the nucleotide bases along its length. So the process begins with the activation of the amino acids, which are going to make up that polypeptide. So what do I mean by amino acid activation? Well, the amino acids in the cell must first be activated before they can be assembled into the polypeptide, and this occurs in two stages. Firstly, the amino acid forms an intermediate with ATP, which provides the energy for the next stage. The intermediate then combines with transfer RNA to form an amino acid complex called amino acyl tRNA. And this reaction is controlled by the enzyme amino acyl synthetase. Now remember that a ribosome's got a large subunit and a small subunit. And a ribosome becomes attached at one end of the messenger RNA molecule. The small subunit attaches first and then the large subunit. And the starting point of the messenger RNA is usually the triplet of bases, which we call a codon, AUG. Now notice how there are two sites in the ribosome. You've got the P site and the A site. This is where the tRNA will move into. So the amino acyl tRNA molecule with the anticodon sequence of UAC moves to the ribosome and pairs up with the AUG sequence on the mRNA. So here you can see the anticodon is on the tRNA and the codon is on the mRNA. These form temporary hydrogen bonds between each other. As the tRNA, which pairs with the AUG sequence on the messenger RNA, always carries the amino acid methionine, polypeptides initially have a methionine as the first amino acid. Then you have a second tRNA coming in. Now, if methionine is not in the final polypeptide, it's going to be removed at the end of the synthesis. During elongation, the ribosome moves along the messenger RNA, bringing together two tRNA molecules at one time, each pairing up with the corresponding two codons on the messenger RNA. And these tRNA move into the A and P sites, which I showed you earlier, of the ribosome. An enzyme joins the two amino acids at the top there, that green square and the purple circle, and the tRNA together, and these form a peptide bond. As the ribosome starts to move over to the next codon, the first tRNA is released from its amino acid, which was methionine, and it's free to collect another methionine from the amino acid pool in the cell. The ribosome moves into the third codon and the sequence of the messenger RNA, so that your second and third amino acids are linked together. then that first tRNA moves out. So it dissociates from the amino acid. So the process continues in this way up to the stop codon. Thus, a complete polypeptide chain has been built up. Now, up to 50 ribosomes can pass immediately behind the first, so that the identical polypeptides can be made at the same time. So a group of ribosomes acting in this way is known as a polysome. So this process continues until the ribosome reaches a stop codon. And these can be UGA, UAG, which you can see here, UAA. These don't attract to tRNA, so it's at this point that the ribosome, the messenger RNA, and the last tRNA all separate, and the polypeptide is complete. Now, sometimes the single chain is a functional protein. Often, a number of polypeptides are linked to give a functional protein a quaternary structure. So what happens to the polypeptide next depends on the protein being made, but usually involves the following. The polypeptide is made into its secondary structure, either by being coiled into an alpha helix or beta-pleated sheet. The secondary structure is folded into its tertiary structure. Then different polypeptide chains can be linked to form the quaternary structure, which will be associated with prosthetic groups. So here you can see how the messenger RNA is translated into the amino acid sequence. So we're going to discuss codons and amino acids in the next topic. Right, in summary, we looked at the different steps of translation. Remember that the amino acid has to first be activated. This uses energy from ATP, and it forms a complex called the amino acyl tRNA. Initiation is when the large subunit and small subunit of the ribosomal RNA bind to the messenger RNA. They bind at the start codon, which is usually AUG. And the tRNA, the first tRNA, usually carries methionine. Then elongation is when the ribosome moves along the messenger RNA, and the tRNA moves into the A and P sites. Peptide bonds form between the amino acids, and then the tRNA is released. And then finally, you've got termination. So the process continues until the ribosome reaches a stop codon, and these are UGA, UAG, or UAA. And these don't attract to tRNA. So it's at this point that the ribosome, the messenger RNA, and the last tRNA all separate, and the polypeptide is complete. And that concludes our lesson. The end.